Well, good afternoon and welcome to another edition of the MTL Doctoral Dissertation Seminar. As you might know, every term, the MTL Seminar Committee selects one of the PhD theses that have taken place in the MTL community in the last year for presentation as a special seminar in our MTL seminar series that I'm sure you know about. And so this is always a very competitive affair with very many strong nominations coming from many different parts of the MIT community that touch or are enabled by research that's going on in MTL. And so today I'm delighted to bring you the awardee of this fall, 2018, who is Dr. Sarvesh Varma. Sarvesh finished his PhD recently, and also he did his master's degree in electrical engineering from MIT. He has a bachelor's degree in nanotechnology engineering from the University of Waterloo. His advisor at MIT in his PhD is Professor Joe Volman, and his research is focused on engineering cell-based sensors and microfluidic systems to study human disease progression. So, Sarvesh, congratulations. We're looking forward to your doctoral presentation.
Thank you so much, Jesus. Okay, so thank you all for coming, and thank you, Jesus and MTL, for this great honor. I'm very privileged to be giving you this talk today about my thesis work. And this work has by no means been just a product of my own effort, but it's really been a product of all of the help and guidance that I've received from so many of you, particularly my advisor, Professor Joel Woolman, and my co-advisor, Guillermo Garcia, who's over there from Harvard. And both of these advisors have been great in not only mentoring me throughout these years, but really helping shape the story that I'm going to tell you about today. And other than people from our group and all of my other colleagues, I have to say that I really owe a lot of my own work here to folks at MTL, and specifically all of you that are here. Not only for helping me with the tricks of the trade in terms of fab, but really creating a positive environment for students like myself to do this research. So again, thank you so much for coming. So I'll begin my talk by talking about a story related to a human disease called atherosclerosis. So this disease is a cardiovascular disease. It impacts the large arterial blood vessels. And this is shown here. And if you look at the causes of death in humans, Western human societies, specifically, let's say here in the US, atherosclerosis has been the leading cause of death worldwide, among other diseases such as cancer, Alzheimer's, diabetes, and so forth. And in fact, this is not only true for the States, but also for many other countries as well. So clearly, it's a very important human disease to be focusing on. And since this is a cardiovascular disease impacting blood vessels, we have to see what's going on there. So just to give you a context, endothelial cells are the cells that essentially form a network of our blood vessels, specifically arteries here. And if you think about what they're supposed to do, they are supposed to contain and circulate blood in our bodies throughout our lifetime. And so to do that, they have to have certain properties. And one of the most important is that they have to resist leakage, like in any flow system, and they have to resist inflammation and thrombosis unless there is a disease or a site of injury. So that is really how they're supposed to behave. But what happens in atherosclerosis is that these cells tend to become dysfunctional. So in other words, they tend to become leaky and sticky to particles such as cholesterol shown here, and they tend to deposit inside the lumen, causing these plaques over time. And these can tend to clog your arteries. So that's how this disease really happens in humans. And if you think about this disease from basically a fluids flow point of view, you have a normal flow that changes very drastically when you have these plaques in your region. And this is just an analogy to show what happens. And you can imagine this tends to increase your blood pressure. And so you might go to the doctor with symptoms of that, chest pain and so forth, and they'll give you some standard recommendations such as, okay, you should do exercise, you should go on a diet and stop smoking, because all of these factors, in fact, contribute to the progression of this disease. And really, these kind of lifestyle changes are very good in terms of maintaining the symptoms. And in fact, you can be fine with these plaques for many, many years in your lifetime. And what actually happens is that these plaques are continuously growing at a slow rate. So at some point, they become unstable and they can rupture. And that can cause a certain clot, and that can lead to a stroke or a heart attack that might kill you. And naturally, that's something you want to avoid. So many of us are actually taking therapeutics for this. In fact, statins are a type of drug that inhibit our own cholesterol production. And turns out about 1 out of 5 people who are older than 45 years old are actually on a statin. And in fact, it has been one of the top selling class of drugs for the past decade or so. So what's important here is that despite the prevalence of drugs such as statins, atherosclerosis remains one of the leading causes of death, suggesting that there must be something else that we have to address and focus on. So if you think about what's going on really in this disease and what we should focus, turns out in the human vasculature, these plaques don't happen everywhere. They happen at certain branch point locations shown here, which can be life-threatening. And we have to focus on what's going on especially there. So you can imagine this is a cartoon of your blood vessel with flow, and as flow comes in, it has a very different profile in the region that has plaques versus the region that doesn't. So even though you have cholesterol particles which are circulating everywhere, they tend to impact a specific region which we call atherosusceptible right here. And so there is something really special going on in this particular region which is unique from that region. And in fact, if you think about the kind of strategies what you might think of, you might want to lower cholesterol particles, so that you can do by statins. But you can't really change the blood flow profiles in all of our arteries. So what is really left is the cells down here. And turns out that in these regions that are atheroprotected, these cells have a very different phenotype, meaning that they are actually behaving biologically very differently. And they tend to be aligned and have a tight barrier function and be in an anti-inflammatory state. As opposed to these cells on here, which we call atheroprone, have a loss of barrier function and are also in a pro-inflammatory state. So and turns out a number of studies have shown that this phenotype actually emerges because of the flow profiles that exist in your blood vessels throughout your lifetime. And in fact, it's just the cholesterol stimulus that then drives disease in this region and not in this region. So thinking again in terms of therapeutics, we realized that we should really be focusing on the endothelial cell phenotype that's happening right here. And if we can target these dysfunctional cells and turn them into these cells, we might have some strategies against the disease progression. Okay, so how would you do this? Typically, if you can think about, let's say in pharma, you might think of using animal models. And that is actually great because they are fully complex physiological systems which have an immune system, the hemodynamics, as well as a hepatic system. So they are quite complex models. But what's interesting is that the relevance of these animal models is not very high. And that's because it turns out atherosclerosis doesn't happen in animals in the same way or in the same rate as it happens in humans. So people have to genetically engineer animals to mimic some of these human diseases. And also the fact is if you want to study endothelial cells, you have to look at the arteries, and therefore you can only do that once in the lifetime of the animal. And so that is not the best way of trying to do either functional screening of your drugs or to learn about biology. And of course, this you don't have so much control over how the disease progresses either. So again, we have learned a lot about the human physiology through these animal models, but they're not great for the goal that we want to do. And so we have to turn towards in vitro models. And then as engineers, we have to think about if we want to model this system, what are the minimum components that we need to have in our device or system? And turns out, your blood vessels and these endothelial cells are exposed to something throughout your lifetime, and that is blood flow. So blood flow, in fact, induces a number of mechanical forces to these endothelial cells, and out of which turns out fluid shear stress is one of the perhaps best characterized and the strongest regulator of endothelial phenotype. So if you, again, wanted to model this system, the two minimum things that you need are both endothelial cells and a way to generate these shear stress profiles. So people go towards these systems such as a cone and plate system, in which you rotate a cone on top of a culture of cells, and the rotation angle and rate can determine the shear stress profiles, or using a parallel plate system in which you drive flow through a circulatory pump, and you can calculate the shear stress by the dimensions and fluid properties. So this is a very standard way of mimicking some of the flow profiles and applying them on cells. But as I said, this is sort of a reductionist approach to mimic what's happening here. So the complexity tends not to be so high, and therefore the relevance to human systems can be moderate to low. However, these are engineered systems, so we have very high control over how we use them, and therefore we can also parallelize and do multiplexed experiments on these systems. So our goal was to really focus on these two aspects, on how do we increase the complexity and human relevance of these models. Okay. So coming back to our original idea, our goal was to look at this and create a device that can mimic the physiological and pathological flow profiles spatiotemporally upon a single endothelial layer like that to model human atherogenesis, or how this disease progresses. And so what are these flow profiles that I mentioned? If you look at, for example, a CFD of a human carotid, which is up here, you'll find that the flows are generally very complex. And if we focus on a region which doesn't develop plaques up here, we can derive some properties. And what we find is that the flows have a pulsatile unidirectional high shear stress waveform, shown here. And the flows have a rotational component, which is helical, shown here. And on the other hand, if you look at the region which is atheroprone, or which develops plaques, we find that the flows have a low oscillatory shear stress waveform, shown here, and the flows are generally chaotic. So together, looking at these medical imaging studies, we can now develop a map of the kind of features that we should have in our system. So then what we do is we turn back to literature and see what actually exists. And as I mentioned, in the macro scale, people have cone and plate and parallel plate systems. And on the micro scale, you can have the same thing compared to the parallel plate when your device is connected to a circulatory pump. And so these kind of studies have, in fact, been very useful to learn a lot about endothelial biology and mechanotransduction. But one of the common aspects of these is that they usually provide spatially uniform flow. So you can't get helical or chaotic flows in these systems. So some people have mimicked this with turbulence, or basically just shaking the culture plate to get fluid motion. And turbulence doesn't happen in vivo, and these models are not very well characterized. And you can't do this in the microscale either. Another limitation is that with a single pump or a single cone, you can only get a single flow profile. So you can either get a unidirectional high shear stress waveform, or you can get a low oscillatory shear stress waveform, but you can't do both. So the fact that you have to pick and choose gives you low throughput. And we know that in microfluidics, for example, by scaling things down, you can parallelize experiments and get higher throughput of experimentation. And in fact, some groups have used valve-based pumping to mimic cardiac waveforms, shown here. And that is very useful. However, typically such pumping can only give you low shear stresses, which are not relevant to the arterial system. So together with these examples and several other studies, we found that there is a lack of systems that can do everything together, and so that became our goal, specifically to provide spatiotemporal, atheroprotective, and atheroprone flows in the same device simultaneously, like it happens in vivo. So with that, we generated a system called an atherofluidic system, which models, as I showed you the cartoon here, in a device. So we have a device which has a left branch that provides atheroprotected flow profiles, and the right one provides atheroprone flow profiles. And we did this in a microfluidic device, and the layout is shown here. This works by 3 modules: a waveform generator, a low oscillatory flow generator, and a cell culture module. But before I actually explain how this works, let's go through how I made these devices. So typically we use this process called multi-layer soft lithography. And this is a standard way of making valved microfluidic devices. And what we do is we take 2 different silicon wafers and pattern photoresists. In this case, the yellow is SU8 and red is a positive one, which we reflow to get the circular profile. And these serve as molds to cast off our devices. So from the bottom one, we actually spin coat an elastomer called PDMS and put that on a substrate here. And then from the top one, we cast off a larger piece and then what we do is we align and thermally bond these two layers together to form our devices. So that's the architecture, and that's shown here. And if you look at this region in particular, in fact, there's an overlap of two channels, and that's interesting. So what we can do is we can connect the bottom channel, which is what we call the control layer, to a pressure regulator. And what we see is that if we pressurize the bottom channel, which is closed at the end, it causes this membrane to deflect upwards. And that essentially closes the top channel. So basically, by the pressure actuation of this membrane, we can stop the flow in the overlying channel. And pressurizing and depressurizing opens and closes these valves, and that's how we can program flows on our devices. So that's sort of the general strategy. And then we can have the device that looks like this with food coloring. You can see two different regions, the red, orange being the flow layer, and the green being the control layer, and the intersection is the valve. Okay, so let's look at the first module, which is the flow waveform generator. So really, this module works to generate the temporal aspects of the atheroprotective profiles. So one of the limitations of traditional parallel plate systems is that if we use external pumps, they are great in providing large shear stresses and volumes, but they tend to be hard to multiplex, and they're very bulky. On the other hand, microfluidic valves can be small and easily multiplexed, but they can only pump small volumes, as I mentioned. So they have complementary advantages, and we wanted to see, can we combine these together or solve this somehow? And our idea was to use valves to modulate, but not to generate shear stress. So how did we do that? We designed a T-junction architecture in which you have an inlet that brings in flow, two cells over here, and you have a bypass outlet which is controlled by this modulator valve. And so if we connect this to a flow source which is uniform in space and time with the valve closed, naturally all of the flow goes to cells. Okay. However, if we open the mod valve here, some of the flow splits to the bypass. Again, standard T-junction operation. So if you look at how this would work, if we have the valve closed, we have a high flow going to cells. Open, we have a low flow going to cells. And essentially, by actuation and deactuation, we can get a flow waveform shown here. But all of us know this is not how the heart pumps blood, right? We have a flow profile which we want to mimic that looks very different than this square wave. So our idea was to go back to the same valve and apply dynamic modulation of the valve position. So really, by opening and closing the valve with a certain dynamic, we can change how the flow splits to cells and therefore get a shear stress waveform. And so now we can get a unidirectional high shear stress flow that comes from one pump and a waveform that's generated by the valves, which can be multiplexed. So we connect our devices to a computer that controls these waveforms and the valves. And this is, again, the picture of a valve which is open with zero pressure, closed with high pressure. And this is typical behavior of microfluidic valves. But in our case, we can apply now a pressure waveform. In fact, one in a sequence of waveforms. And what we see is that in response to this, we get a valve that essentially beats in response to these pressure waveforms. So it's not only digital on and off, but it responds by changing its position in analog states. So now, taking this beating valve, we can go back to the device and see how it affects flows. So what we can do is flow in fluorescent beads through our device, and you'll see two videos. On the left, you see a protective waveform, which is applied at, in this case, low shear stress, so you can see the pulsatility, and on the right with constant flow. And we can do this at 80 beats per minute, physiological range. And we can also do this at higher heart rates. So for example, if you just did exercise, your heart rate goes up, and we can mimic that waveform as well. And then I can take these videos and do PIV to quantify the flow profiles. And what we find is that against the human reference, which is shown in gray, we can very closely match the waveforms in our device to what happens in humans. And we can do the same thing at higher heart rates as well. So the waveform modulator can successfully generate physiological waveforms that happen in humans. Okay, so now let's look at the other module, which is the low and oscillatory flow generator module right here. And that is basically to generate the dynamics in the atheroprone region. So this basically works by two configurations, one shown here with two oscillatory valves, 1 and 2, and an inlet and a flow resistor. Okay, so if we apply flow, what happens is before applying flow, we can close one valve, in this case valve 2, apply flow, and flow goes forward, up and out in the left branch. And in the right branch, what it does is it also goes forward through the resistor, up and out. So the flow directionality is forward, and the magnitude is lowered by this resistor. It looks like that. And in the flip configuration, we toggle the valves. Okay, so we turn off the valve that was open before. In this case, we close the top one and apply flow again. What we find is flow is forward, up, and out in the left branch again, like the left case. However, the right branch can't do the same thing as the left branch. So it goes down, up, and out. And so what we have done essentially is by toggling the valve states, we create flow reversal specifically in the atheroprone or in the right branch, right here. And that doesn't affect the left branch flow directionality. So we know that in vivo, flows are low oscillatory shear stress in the atheroprone region. And so we can get a resistor that lowers that shear stress and the valve toggling that gives us that oscillation. So how do we actually measure this? There's a metric called OSI, which is for the relative time spent in reversal. So if I walk straight forward, that is OSI of 0. If I take one step forward, one step backward, that's OSI of 0.5, so purely oscillatory. And for patients, it tends to be in the range of 0.3 to 0.45. So if we look at the valves, we can now program how these valves are switching over time, therefore controlling the OSI or the flow reversal in our devices. And really, this allows us to get a low shear stress that comes from the resistor and an oscillatory waveform that comes from these valve actuations. Okay, so how does this work? We can again look at bead flows in our device. This is in the right branch, which is atheroprone. We see that by using our valves, we can generate at the same heart rate and shear stress flow which is, has an OSI of 0.25 with the waveform shown here. And we can increase this to a OSI of 0.4, which is nearly stagnant. So the flow doesn't move that much. And in fact, not only we can tune these OSIs, but what we can do is we can further take these programs and tune them very closely to what happens in human patients. So if you look at your own plaque region, you'll get a waveform which is very similar to the gray waveform shown here. And we can match that very closely in our devices with the red waveform. And again, these are tunable waveforms. So giving us the ability to now program in the kind of atheroprone waveforms that are happening in humans in our device. And then also the other advantage is that this can be applied at on-demand heart rates, as we can change that. And also it can work in conjunction with the atheroprotective waveform generator that I showed a couple slides ago. So there's completely two different ways of controlling the dynamics of one region and another region. Okay. So, so that's on the dynamic side. What's important is that as I told you in the video, that the blood flows are very complex. They have a spatial component to them, right? So they don't just flow straight. And turns out one of the first people to describe these spatial flow properties was Leonardo da Vinci. And so some 500 years ago, these are his own lab notes written in reverse Latin, where he describes the flow disturbances as flow leaves the heart and goes to the aorta. So he was very interested in flow swirling, helical flow, and vortices in his lifetime. And he describes this for the first time over here. And many, many years later now, we have sophisticated tools that can actually measure this and quantify how these flows might be happening in humans. So this is a 4D MRI image showing from the human heart how these flows have a rotational component as it goes down. And in fact, you know, we can quantify these helical flows and flow circulations, but this is not exclusive to the human aorta. In fact, it turns out that these flows happen in in many different regions of our vasculature, different arteries, and also it's conserved in many other species. So this is a very unique property of the flow, which in fact exists and is conserved. Coming back to how we can model this, again, as I said, there are not many ways to do this in vitro. So as I said, people tend to shake their culture plate. Okay, that might create some fluid motion, but it's nowhere similar to what happens here. And so therefore, our idea was to look at this context and think about how can we mimic these flow properties that are happening inside of you? So we went back to Microfluidics 101, and we used this architecture of channels having grooved ceilings. And in fact, turns out this has been around for a very long time and used by many groups, including our own, to mix liquids inside our devices. So if you use these grooves, then they tend to create transverse flows, and that can be useful for mixing flows in the microscale. What we can do is take this idea and now design specific grooves that can now give us the spatial profiles of what are happening in our blood vessels. So we design channels, in this case, with slanted grooves that, with the flow simulation showing the particle trajectories, can actually induce helical flow. And you can see that more clearly in the simulations down here on the left. And the bead flow profiles in our device also show an anti-clockwise rotation of the fluid inside our devices, suggesting that we can really now get these helical flows in our devices. We can further go ahead and change the architecture into staggered herringbones, and that gives us chaotic flows, which are a feature of the atheroprone region, where you get plaques. So you can see again by the simulation, the particle flows, they're very different from the helical flows generated by the other grooves. So literally, by superimposing these grooves on top of our channels, we can now superimpose the spatial aspect on top of the dynamics that we were controlling with the valves earlier. So that allows us to now bring in the spatial properties on top of the temporal dynamics. So how does this all come together? If we look at the architecture that we wanted to mimic, we can now have devices which have two distinct regions. This is all connected in the same device. And what you'll see on the left is that we can get a high shear stress pulsatile waveform. You'll see these beads pulse. And that is shown by the fast motion or the blurring of the beads as they go forward. Versus on the right side, we have an oscillatory low shear stress waveform, as I mentioned. And these can be provided simultaneously and can be programmed. And furthermore, we can do this with grooves in our device, and this is at real time. We can get, for example, the unidirectional and multidirectionality that happens in vivo. So by connecting all of these modules and designs together, we can now get the full map for the first time in our device. So we can get the spatiotemporal aspects of both these features. Okay, what is important is that not only we can do this technically, but we can apply this on cells. So let's look at the cell culture module, where we can culture human cells. And just to give you reference, again, this is what our device architecture looks like, trying to model this scenario. We have a left branch that provides the atheroprotective region and flows, and a right branch that provides the atheroprone flows on these cells. And this is all a continuous device, so we can do this on a continuous layer of cells. And so what we can do is we can take primary human endothelial cells and then culture them in our device. So these are two branches, the left branch and the right branch, and you can see them in the phase contrast and probably a bit better with the stained nucleus in fluorescence. So what we can do is, in fact, we can put these cells in our device, grow them over time, and monitor them under flows. So we can really see how they're changing in response to these flows. What we can do further is, in fact, after the end of the experiment, we can use branch valves to take them out. So we can close a branch valve. I can dissociate these cells and then wash them out. So now I've selectively taken these cells without touching these guys, and then I can do the same operation here and recover these cells. So you can essentially think of this as doing like a microscale surgery on the cell culture that we were doing, studying for a long time. And this is important for a couple reasons. As I mentioned, that not many devices give you this functionality of applying and mimicking these flow profiles on the same endothelial. In fact, none of them do. And but what's interesting is that this is hard to do also in vivo. So if you had an animal that you wanted to study, you would have to cut out that tissue, cut out that blood vessel, estimate where these regions would be, for example, red being atheroprone and green being atheroprotected, and then take out those cells, and then you start all over again with a different animal. So you don't have a lot of cells to work with, and you can only do this sequentially or with one animal at a time. And in our devices, we can use human cells. And we can scale this to have many more cells to work with. And we can very specifically take them out for any off-chip assays. So we're not limited to imaging only. And that's a unique advantage of the cell culture module. So let's look at the big picture again. What we can say now is that with our devices, we have high complexity in the flow generation and control. And in terms of experiments, we can also not only do imaging, and we can take the cells out to do off-chip assays. And being in the microscale, we can think about making parallel devices and architectures to increase the throughput and so forth. But one thing that is very important to people like myself or anyone else doing research of this kind, where they're making devices to mimic something in humans, the toughest question is the human relevance one, which is to say, whatever you're doing, is that really representative of what's happening in humans? And so to answer or to address that kind of issue, we have to really turn towards biology and see what's happening in the body and what can we see in our devices. So for that, we chose cells which were called human umbilical vein endothelial cells, or HUVECs. And these cells essentially come from moms. So when moms deliver babies in hospitals, they take out the umbilical cord, they take out the endothelial cells, and we can use those cells to study vascular biology. And in fact, these cells, or this method, is very common to study this kind of scenario in the vascular biology community. So it's a very standard cell choice, and we basically took the same cells from our collaborator, Guillermo. And the context of what we wanted to study was the onset of endothelial dysfunction. So as I mentioned, the flows can actually condition these cells very differently in the different regions before you even have cholesterol or before the plaque starts to happen. So we wanted to study that first stage of how these flows are impacting the cells as the first step in the disease progression. So taking these cells in the context of atherogenesis, we wanted to see what actually happens biologically. So our collaborator Guillermo's lab actually was one of the first people to find and quantify the waveforms that happen in the human carotid. For example, the region which is atheroprotected has this high pulsatile shear stress waveform versus the low oscillatory waveform shown here in the atheroprone region. These are the same waveforms we're mimicking in the device. Okay, so what they also found was if they take the tissue out and look at the endothelial cells, the region which is atheroprotected have a very high level expression of this protein called KLF2, and that's shown in purple here. If you look at the same staining in the atheroprone region, it's absolutely gone. So there's no staining of KLF2 or production of KLF2 in that region. So they were interested in this, and then what they did was they took endothelial cells, HUVECs, in a cone and plate system and applied an atheroprone waveform, which happens in the blue region. And what they found was there was little to no expression of KLF2 again. But if they changed the waveform into an atheroprotective one, they got very high induction of KLF2, suggesting that KLF2 responds specifically to the atheroprotective flow waveforms that happen in humans. And they can actually recapitulate that in human cells in vitro. So since then, their group and several other groups found that KLF2 is not just a random gene in endothelial cells, but it's actually a critical regulator of endothelial health. And in normal physiological conditions, it actually regulates formation of new blood vessels and controlling vascular tone. But in the context of atherosclerosis, it drives the endothelial cells into an anti-inflammatory, antioxidant, and antithrombotic state, which leads towards an anti-atherogenic phenotype. What that really means is that if the cells have high level of KLF2, they will be protected from the disease. If they have low levels of KLF2, they will be more prone to disease. So with that, they realized that KLF2 is a very important biomarker for endothelial cells, and they generated a sensor in HUVEC cells. Essentially, the idea is that by taking the regulation and the promoter of KLF2, you can drive green fluorescence, shown in this cartoon here. So if the cells are exposed to an atheroprotective waveform or flow profiles, they will have high levels of KLF2, which drives high level of green fluorescence. And in the opposite case, if you have an atheroprone stimulus, you'll have low levels of KLF2 and low levels of GFP. So as a method, we can now look at fluorescence to estimate or get an inference about what the KLF2 levels will be to then infer about what the endothelial phenotype would be. So it becomes a method that we can use in our devices to start looking at endothelial biology. So we took these sensors in our devices, and these are just sensors under flow. And this is the morphology at time 0. I apply flow from the right to the left, and you see the cells start to behave and respond very differently when the flow is applied. And you tend to see over many hours that these cells become elongated and align in the direction of the flow. And if you look at steady state, for example, something like this, these kind of alignment and morphology is very similar to what you would find if you cut open just a blood vessel in an organism and just look at it. So these steady-state morphological profiles match very closely if you apply the atheroprotective flow profiles. We can also do this now with the sensors. So not only looking at the morphology on the left, we can do real-time imaging and look at the green fluorescence over time. So this is after 3 days, we can continuously monitor the green fluorescence. And what I hope you can appreciate is that the green fluorescence really starts to turn on and go to a higher level compared to the time 0 case, suggesting that if we apply the atheroprotective flow stimulus, it kicks on the KLF2 production, and then we can see that through our green fluorescence in our channels. So then we can also go back to our own results and look at time 0 versus time, let's say, 24 hours, and not only look at morphology, but also the sensor signal, which we can quantify as a metric to see how the cells are doing in our devices. And then if we also did the opposite experiment, that if we apply disturbed flow, or in this case, atheroprone flow, we get no alignment of cells, and these cells look very similar to what atheroprone cells look like, and we get no green fluorescence. Again, that's representative of the low KLF2 levels that exist in the atheroprone region. So together, by these kind of experiments, we can get both the morphological signatures and as well as infer the phenotype through this KLF2 GFP sensors in our devices, which give us confidence, because really these are the kind of phenotypes that people have seen in other labs and other models. So we know that we can now begin to use the same sensors in our devices, and our devices can now be used to ask new questions. So we went back to the map and saw what can we actually study. And turns out there have been a lot of studies that looked at the atheroprotective or atheroprone waveforms. And we have learned a lot about those responses. But one thing that's specifically unique in our devices is the ability to apply a helical or chaotic flow. And since there have been no other models to do that, we can really start to apply those flows and see how the cells are responding differently. And that was the focus that we wanted to really get into with our system. So one of the first things that we did was we can, as I said, apply helical flow to specific grooved architecture. So this is how the cells look like without that stimulus, basal green fluorescence. And if we apply helical flow in these grooved channels over 3 days, what we find is the cells tend to align helically in our device and express high levels of green fluorescence, or KLF2, in these conditions. So that is very unique, because these kind of experiments, again, have not been done in any other system, and we can really start to measure and observe how these phenotypes are emerging in response to helical flows. So that's just one marker. Turns out there's another marker called NF-kappa B. And that is a marker that doesn't protect you from disease, but in fact promotes disease. So it's the flip side of the phenotype. And in fact, if you look at the staining for this protein in the cell, what you find is that if you stain the cells and you find high staining or white color inside the nucleus region, shown here, the cells are actually in an inflammatory state. So this protein goes into the nucleus and drives many genes which initiate inflammation. However, if you find staining which looks like this, so you get bright staining outside the nucleus, you know that these programs are actually off, and the cells are in an anti-inflammatory state. Okay? So if you look at in vivo, in normal animal arteries and human arteries, what we find is that in the atheroprotective region, the profile looks very much like the cartoon here, that the cells are in an anti-inflammatory state when you give them those protective flow profiles. And if you look at the cells in our device, it looks very similar to what happens in vivo, both in terms of morphology, but also in terms of the NF-kappa B profile, that it being cytosolic suggests that the cells are indeed in an anti-inflammatory state like they are in vivo. So by KLF2 and NF-kappa B, we can get at least 2 features, and in fact, we can extend this to many other biomarkers and other studies to really see how these phenotypes are emerging in response to our spatial flows. So that's just one example. We then went back to the map and designed a number of other studies which looked very broadly at the impact of spatiotemporal flows. So we compared what happens if you have high shear stress versus no flow at all. Then in high shear stress, we compare the impact of dynamics, so constant flow versus the atheroprotective waveform, and then chaotic, helical, and uniform flows in each of these profiles. So what's the impact of the spatial flows within these dynamic conditions? And then we used the same sensor, so the green fluorescence readout, to get an inference about how the cells are responding. And we can use that to quantify the relative impact. So if you look at the no flow control condition, this is how much the cells respond if you have basically no flow. And if we apply high shear stress flow, all of the sensors respond high. And that's not surprising because, as I said, KLF2 is indeed induced by high shear stress flows. But now let's try to focus on a subset of the conditions. So if you look back to our map, this is the atheroprotective map, we asked a question that, okay, what if we take away the waveform, right? And if we change the spatial profile from helical, which exists in atheroprotective regions, to chaotic, which happens in atheroprone regions. So looking at the impact of the spatial aspect in the context of high shear stress. And what we found was that through our sensors, we found an increase of the KLF2 GFP signal, or the sensor response, when we had helical constant flow compared to chaotic constant flow. And I have to say these results are sort of counterintuitive to us, because we didn't think the cells could in fact respond to what the bulk flow profile is. But they were indeed responding differently in this case. And the second important thing is that we found that the helical flow, which as I mentioned was found in the atheroprotective regions, tends to enhance KLF2 expression, as opposed to chaotic flow, which happens in the atheroprone region. So if we take, quote unquote, the bad flow property and apply it to these cells, even though they have high shear, it showed us that it gave us lesser expression of KLF2 compared to the spatial profile that they should be seeing, which is helical flow. Then we looked at a different question. Okay, let's keep the chaotic flow. What happens if now we add on the temporal dynamics? So what changes if we have the waveform now? And what we found was, again, that if you kept the chaotic flows, which are atheroprone feature, and you added the protective waveform, you again get an enhancement of KLF2 signal, meaning that the waveform can somehow rescue or enhance KLF2 despite being in the chaotic region. So it has something unique that can enhance that response. And then finally, we asked the question that if we kept the helical flow and also added the waveform, so both the positive stimulus, what happens? We found was, in this case, we saw no change. So that could either mean that our sensor response was saturated, or that there was no synergistic response when you have both the features of the waveform and the helical flow. So we did these experiments and several others in the atheroprone map and compared the relative response when you have the waveform and the spatial, and we found very distinct responses in terms of the helical and chaotic flows at the very least, and also other hypotheses. So I wanted to say that with these sensors and our devices, we can get very unique insight about these helical and chaotic spatiotemporal flows might be differentially affecting endothelial cells. And I hope you can realize that the sensor and this gene is only one gene, or only one kind of sensor that we're using. But it might be the case that, in fact, there's a number of other changes going on that we don't just see here, and we can, in fact, use our device to, in fact, explore those other assays as well. So coming back to the big picture, I should say that, in fact, one of our inspirations to mimic these helical flows came from the medical imaging community. So the CFD community had been telling us that helical flows are actually found in atheroprotective regions. So they took a lot of images of healthy humans and found that if they have helical flow shown by these two colors, they have a very small plaque-prone region, which is here. In other patients, or actually human subjects, they found if you have a loss of this helical flow or multidirectional flow, you have a much larger plaque-prone region. So just purely by correlation studies, they told us that helical flows might actually be a surrogate marker, or some sort of marker that you can look for in the imaging domain to infer the atheroprotective profile. In a very different community, which is the graft and stent design community, people found that if you induce flow rotation, for example, through grooved stents or through actual stents which are helical, people found that in animals you can, in fact, have, in this case, with a straight stent, a narrower opening of the artery compared to helical stent, which was larger. So a very different community telling us that there might be something going on with helical flows at the macro scale, right? Very correlation-based studies. But with our device that can model what happens in humans, we can now look at a very different scale, which is on the cells. And so specifically by using the cell sensors, we can now generate the first few hypotheses on if and how these helical flows are actually acting as a physiological stimulus to the cells. And chaotic flows might be a pathological features. So now we can really extend these studies to the vascular biology community for the first time that can study the impact of these spatial flow profiles that they could not do previously, and then do that on the devices. So that's one of the novel enabling aspects of our technology. And of course, this is not the only thing you can study. It turns out that we can look at these mechanics and transduction mechanisms over time, but extend this to examples such as personalized medicine, in which case we can take cells from a patient and apply their own flow profiles, so do personalized responses, or do tissue engineering to look at later stages of the disease with multicellular components, or go back to the original problem, which is to say, how can we use now this device that can mimic these flow profiles to start thinking about cardiovascular drug screening? So really going back to the clinical problem to think about, can we use this now or generate newer scaled models to discover new drugs that could be useful for treating atherosclerosis? So with that, again, I would like to thank you all for coming. And again, thanks to my advisors, Professor Joel Woolman and Guillermo Garcia, who have really helped me shape this story. And again, all of you, my friends and family, for coming. And also, I'm very grateful to have this honor to give this talk to you. Thank you.
Thank you, Sarvesh. I didn't mention at the beginning that one of the criteria for selecting the awardee of the doctoral dissertation seminar is the ability to give a compelling and engaging and inspirational talk to a wide range community. And I hope you can see that Sarvesh has done just that and more. This was really a very beautiful talk on a very complex topic, but you've done it in a way that it's been a real joy to understand and to learn so much about such difficult things. So we can take some questions now. I have one, and I apologize in advance for its naivete, but you started telling us about cholesterol, something that we all worry very much about as sort of the underlying issue behind this problem. But I didn't see you pumping cholesterol through these channels. Why not?
Is that's a great question.
Difficulty in seeing how does the cholesterol respond to all these different flow issues and waveforms and so on?
No, that's a great question. We were in fact caught by our own curiosity by the earlier effects that we were seeing in terms of the response to the spatial flow profiles. So it turns out that indeed we can flow cholesterol and see how the cells are responding differently in our devices. And there's nothing different in terms of the experiment that you would have to do to look at that. However, what we wanted to do was, as I sort of inferred to, was that atherosclerosis is really a two-hit model disease. So you have your blood flow profiles, which are unique to all of us, and those flow profiles condition the cells to be either protected or susceptible to disease. And then you add cholesterol, which then drives the disease, specifically in that region. So we wanted to look at the first stage, which is the conditioning of the cells and how they might be responding differently, because now we're adding the spatial component, which hasn't been explored. So we want to see if that spatial component indeed is that conditioning the cells differently. And we found with our results that the helical flow and chaotic flow, which are the two different spatial properties, are impacting the sensors in a way. So that was our original focus. But as we move beyond that and look at more characterization of the cells, we can then add cholesterol to see is there a difference in how these cells drive disease in our device in those two regions. Yeah.
Very good. All right, let me take some questions. And please wait for me to reach you with my microphone so we can record your question.
Yeah.
Very nice talk.
Thank you.
So one question I have, it looks like one of the technical challenges would be trying to establish confluent monolayers and having good morphology of the endothelial cells. And some of the things you're doing to create the spatial flow patterns seem to go against that a little bit. So I'm wondering, my recollection is that for chaotic mixing, the chevrons are on the order of 20 or 30% of the channel height to get effective mixing. So how do you deconvolute the effect of the flow from the effect of the feature? For instance, the chevron versus the straight groove. That's going to affect the cell culture versus just the flow against those endothelial cells. How do you deconvolute things like that?
So that's a good question. What we found was, in terms of maintaining endothelial culture and morphology and confluence, as you know, these are cells that like to be very close to their neighbors. And if you keep them far apart, they don't like it. So that's kind of like a disease state. So in our devices, we had to make sure that, in fact, when we develop the cell culture protocols, the cells are indeed happy and confluent and look like they do in vivo. So we did those modifications and optimization first from the straight channel, and then also did that in the helical and chaotic channels. And so our protocols, in fact, we don't have to change that. Our protocol, in fact, helps us to maintain those cultures in our devices for many days, which in fact is a challenging thing to do, especially at high shear stresses. So we didn't find that we had any, I guess, drastic effect on the endothelial culture because of the grooves. And we could still continue with our experiments with that aspect added on.
Yeah, thanks again as well.
Sure.
I just had a question if you knew, just in your research, if there's a size of blood vessels that preferentially forms plaque, and kind of what the overlap of that size blood vessel, what that overlaps with your microfluidic devices.
So that's a good question also. So the question is there a specific size where the disease happens or not? It turns out it's, as far as I know, atherosclerosis is a disease of the large blood vessels, and large is used as a term, but we don't really quantify what large means. In fact, it doesn't happen in the microcirculation or even in veins. So the understanding is that it's the regions where you develop these flow profiles, which happen to be in large arteries. So in terms of our own device dimensions and scale, at least the human aorta is something between like 15 to 30 millimeters in diameter. And our devices, at least for now, are about 0.5 millimeter wide. So we can, but the good thing is that we can scale this. So if we wanted to look at the full range, we can, or have more cells, we can do that. But the important part is to mimic the spatial flow profiles and temporal waveforms, which we do at that scale. So I hope that answers somewhat of the question. Yeah.
More questions?
Yes.
So, really nice talk. So I have a question. So you use the grooves to mimic the spatial profile of the blood vessel.
Yeah.
Is it a real case that the blood vessel has the grooves inside?
No, of course not, right? I mean, if you,
But
sorry.
how it naturally happens in the human body? Like, why doesn't, human body does not, if blood vessel does not have the grooves, how does the profile happen in real case?
That's a great question too. So if the blood vessels don't have grooves, how do they get helical flows at that scale, right? And the answer is that the fluid flows are very complex, both in terms of the Newtonian or non-Newtonian mechanics, and also in terms of the actual velocities and the scale of the flows. You generate, for example, sometimes inertial effects of the flow, or the for example, the curvature or the 3D nature of these arteries, in fact, induces some of these rotations. So, in fact, just for the aorta itself, it looks like, kind of like you can imagine an umbrella handlebar coming out of a heart, right? But that's just a cartoon. It actually doesn't look like that. It has a curvature to it too, which we don't know why it exists or maybe why it was conserved. But some of that is at least thought to have playing in a role in inducing these spatial effects. So there's a lot of structural and, I guess, dimensional reasons why these spatial flows emerge in an animal or in a human. But we just wanted to take those properties as a map and then mimic that in our device. Yeah.
Any more questions? Okay. If not, thank you again for a wonderful presentation. Please don't go away. We have to give him his award. So, Sarvesh, this is your
Thank you so much.
presentation award for the fall of 2017.
Thank you.
Always great.
Yeah, thank you so much. I really appreciate it.
Do we have a poll? We don't have a poll. Or you can take us a picture.
Yes, sure.
And Joel, I'd also like to add you to